Hello students, welcome to EPG Pathshala. My name is Dr. Priyanka and I teach Anthropology at the Department of Anthropology, Dr. Hari Singh Gaur University, Sagar. Today we are going to discuss about allelic frequency, genotypic frequency and Hardy-Weinberg law in this module. This module comes under the paper of human population genetics. So let's see that what we are going to learn in this module. This module aims to understand natural selection as a blinding process of one of the microevolutionary forces. It aims to understand various components, modes, and types of natural selection. We will be understanding various case studies of natural selection in human population. So let's begin this module. Before we proceed towards Hardy-Weinberg law, we need to know about population genetics. Population genetics is a field of biology that studies the changes in the genetic composition of a biological population. The change results from operation of various microevolutionary factors, including natural selection, drift, gene flow, and the various other processes. These evolutionary forces shape the gene pool of a population. A population evolves through change in the gene pool. Population genetics is the area of research that examines the allele frequencies in a population and attempts to identify the various factors that can cause allele frequencies to change in a specific group over a period of a time. A population is a group of interbreeding individuals that share a common gene pool. By the term gene pool, we means a total complement of genes shared by the reproductive member of a population. Once human populations have been identified, the next step is to find out what evolutionary force, if any, are operating upon them. To determine whether evolution is taking place at a given locus, population geneticists measure allele frequency for a particular trait and compare these observed frequencies with the set predicted by a mathematical model called as Hardy-Weinberg equilibrium equation. Evolution or change in the allele frequency thrive on the variation. Therefore, genetic polymorphisms are maintained at the species level. In short, population genetics is a study of frequency of alleles, genotype, and phenotypes in a population from a microevolutionary perspective. The goals of population genetics are pursued by formulating mathematical models of gene frequency dynamics and extracting the conclusion to study genetic variation in the actual population and then testing the conclusion against the empirical data. Before we proceed to understand Hardy-Weinberg law, we need to remember three important terms. One is a Mendelian population, because population geneticists mainly focus on Mendelian population. Let's learn more about genotypic and allelic frequencies. An obvious and persuasive feature of life is variability. Variability can be seen in a group of a student as no two students in a class are similar in a typical college class, the members of which vary in eye color, hair color, facial feature, blood types, susceptibility to numerous diseases and disorders. Humans are unique in their extensive variability, and much of this phenotypic variation is hereditary. Recognition by this phenotypic variation and its extent to potentially adapt to environment led Charles Darwin to deduce the idea of evolution through natural selection. In fact, even more genetic variation exists in population than is visible in the phenotype. Much variation exists at the molecular level, owing in part to the redundancy of the genetic code, which allow different codons to specify the same amino acid. Thus, no two members of a population can produce the same protein even if the DNA sequence are different. An important but frequently misunderstood tool used in population genetics is the mathematical model. They are oversimplified representation of a process and effects between the factors. How to calculate a genotype frequencies? A frequency is defined as a proportion or a percentage, usually expressed as a decimal fraction. For example, if 20% of the alleles in a particular locus in a population are A, we would say that the frequency of A allele in the population is 0.2. In a large population, a sample of frequencies can be calculated for this sample. The genotypic and allelic frequency of the samples are then used to represent the gene pool of the population. The genotypic frequencies are calculated by simply adding up the number of individuals possessing the genotype and then dividing it by the total number of individuals in the sample, which is denoted by the letter N. Thus, for the locus with three genotypes, namely capital A, small a, and small a, the frequency of each genotype is frequency for capital A can be calculated by dividing the number of individuals of capital A genotype by the total number of individual samples. Similarly, the frequency for capital A small a can be calculated by dividing the number of capital A small a individuals by the total number of individuals in the sample. Likewise, the frequency for small a, which can also be denoted as homozygous recessive genotype, can be calculated by dividing the number of small a individuals with the number of individuals in a sample denoted by letter N. The sum of all genotypic frequencies should always equals to 1. Now, how to calculate the allele frequency from the genotype frequencies? The gene pool of a population can also be described in terms of allele frequencies. There are always fewer alleles than the genotypes. So, the gene pool of the population can be described in fewer terms when the allele frequencies are used. In sexually reproducing population, the genotypes are only temporary assemblage of the alleles. As genotype break down in each generation, when individual alleles are passed to the next generation through gamete and make up the gene pool. Or in other words, we can say genotype is a combination of different alleles. Allelic frequency can be calculated by two ways. It can be calculated from the numbers of genotypes, or it can be calculated from the frequencies of the genotype. To calculate the allelic frequencies from the number of a genotype, we should count the number of copies of a particular allele present in a sample and then divide by the total number of alleles in the sample. The formula for this can be displayed as frequency of an allele is equal to number of copies of an allele divided by number of copies of all the alleles at one locus. For a biallelic locus, that is, for a locus with only two alleles called as capital A and small a, the frequency of alleles are usually represented by symbol P and Q. Based upon the number of genotypes, they can be calculated using the following formula. The frequency P, that is, frequency of A allele, can be taken as twice the number of individuals of AA individuals with the number of capital A small a individual. This sum can be divided with twice the total number of individuals in the sample. The same can be done for the another allele. As we have mentioned that the allele frequency can be calculated by either number or the frequency of the genotype. Now we will see that how to calculate the allele frequencies using the frequencies of the genotypes. The sum of allelic frequencies from the genotype frequencies, we have to add the frequency of homozygote for each allele to half the frequency of heterozygotes allele of each type. We obtain the same values of P and Q whether we calculate the allelic frequency from the number of genotype or from the genotype frequencies. Population is an interbreeding group of organisms that share a common gene pool. By the term of gene pool, we mean that collection of all the alleles in the population. Evolution is understood as the change in the allele frequency in a population. A non-evolving population is one where there is no change in the allele frequency over a time, and if there is a change in the allele frequency, we say that the evolution is operating in the population at that particular locus. The foundation for population genetics was laid in 1908 when two people, one G.H. Hardy, an English mathematician, and W. Weinberg, a German physician, independently published their work, which later became famous as Hardy-Weinberg equilibrium. It was the outcome of raging controversies that shortly developed after the rediscovery of Mendel's law in 1900. One of the main questions at that time was regarding the possibility of early elimination of recessive allele from the population by virtue of its recessiveness. G.H. Hardy and W. Weinberg independently demonstrated the folly of this argument. They showed that randomly mating large population would be expected to retain the allelic variation by simple Mendelian principle unless some other forces acts on this variation. The basic approach in population genetics makes use of the mathematical model developed by them, which is known as Hardy-Weinberg equilibrium equation. The Hardy-Weinberg theory of genetic equilibrium postulates a set of conditions in a population where no evolution occurs. In other words, Hardy-Weinberg law presents a hypothetical condition by which a population would be assumed to meet conditions such as infinitely large size, occurrence of no mutation, occurrence of no selection, randomly mating, and no gene flow happening in the population. If all these conditions are satisfied, allele frequency will not change from one generation to the next generation. However, remember that Hardy-Weinberg equilibrium are just that, an idealized hypothetical state. In real world, no actual population would fully meet any of these conditions. If the observed frequencies differs from those expected by this model, then we can say that the evolution is taking place at the particular locus in the question. And if it is not the case, that means no evolution is occurring. The Hardy-Weinberg equilibrium makes several simplifying assumptions about the population and provides key predictions if these assumptions are met. The assumptions can be if the population is large, random mating, and not affected by mutation, natural selection, then the prediction is that the genotypic frequencies stabilize after one generation in the proportion p square, 2pq and q square, where p equals to the frequency of allele A and q is the frequency of allele A. P square is the frequency of AA genotype, 2pq is a frequency for AA, capital A small a genotype, and q square is a frequency of recessive small a genotype. A large population of sexually reproducing organism is considered where the organisms are assumed to be diploids. By diploidy, we means two copies of each chromosome, one received from each parent, is present. The gametes produced by them are haploid, that is, only one copy if each chromosome is present in the gamete cells. Two haploid gametes fuse to form a diploid zygote during sexual fertilization, and then it grows and develops into adult organism. For an autosomal locus with two alleles, there are two possible alleles, A1 and A2. Organisms with A1A1 and A2A2 genotypes are called homozygotes. Those with the A1 and A2 genotypes are called as heterozygote. The relative frequency of the three genotypes in the overall population may be denoted as frequency of A1A1 genotypes, frequency of A1A2 genotype, and frequency of A2A2 genotype respectively. The sum of these three frequencies of these three different genotypes should be equivalent to 1 and are same for both males and females. The relative frequency of A1 and A2 allele in the population may be denoted by P and Q, where P plus Q should also be equal to 1. For a locus with 5 alleles, there is threshold between 5 allele frequencies and 15 possible genotypic frequencies. This simplification is particularly useful for a locus with 2 alleles because it allows us to follow change in the frequency of one allele instead of frequency of 2 or more genotypes. The relationship well known for Hardy-Weinberg equation is P square plus 2PQ plus Q square is equals to 1, where P and Q are allele frequency for a genetic locus with 2 alleles. If we consider a diploid organism, there are 3 possible genotypes in the population: A1A1, A1A2, and A2A2 that are present in the frequencies with P, H, and Q respectively. The sum of genotype frequencies for A1A1, A1A2, A2A2, which we are seeing as PHQ, should also be equal to 1. Because all the alleles in the two homozygotes A1A1 and A2A2 are A1 and A2 respectively, and half the alleles in the heterozygotes are A1 and half are A2, the allele frequencies in terms of the genotype frequencies are then small p is equals to p plus half of h, where p is a frequency of genotype A1A1 and h is a frequency of genotype A1A2. Small p is the symbol used for allele frequency of A1 allele. Similarly, for Q allele, we have the sum of frequency of genotype A2A2 with half of the frequency of genotype A1A2. In random mating between individuals of 3 genotypes, 6 mating types can be observed. These 6 mating types, their frequencies, and the expected frequencies of their offspring genotypes assume that gametes segregate in Mendelian proportions. For example, the mating between A1A1 genotype and A1A2 produces half A1A1 and half A1A2 and so on types of gametes. The graph given in this slide demonstrates the Hardy-Weinberg principle assuming random mating in the parents and Mendelian segregation to produce the progeny. As we can see in this chart that we have individuals with 3 different genotypes, namely A1A1, A1A2 and A2A2. The frequency of these three different genotypes can be represented by the letters P, H, and Q respectively. Random mating between them results in 6 types of mating, which is given in the first row of the chart. Now let's try to understand this chart properly. In first type of a mating, where A1 individual mates with another A1 individual, all the progeny is of A1 genotype. Since the frequency of A1 is represented by the letter P, therefore the frequency of the progeny can also be written as P square. This is same for the mating type where A2 individual mates with A2 individual, producing Q square frequency for the progeny, because all the genotypes in the progeny are of A2 type. The Hardy-Weinberg law holds true for any frequency of A and A as long as the frequencies add to 1 and all the assumptions are evoked. A population in which the allele frequencies remain constant from generation to generation and in which genotype frequencies can be predicted from the allele frequencies is said to be in the state of Hardy-Weinberg equilibrium for that locus. Genotype proportion may deviate from Hardy-Weinberg expectation for several different reasons. The most significant evolutionary factors are selection, inbreeding, gene flow, and thus it is often said that Hardy-Weinberg proportion are expected only in the situation in which there is no selection, random mating, and gene flow. If the equilibrium gets disturbed, that shows that the forces of evolution are working on it. So far we have seen only 2 alleles, 3 genotype system. The Hardy-Weinberg principle can also be applied to X-linked genes and to the genes with multiple alleles. For an X-linked genes, such as the one that controls color vision, the alleles frequencies are estimated from the frequencies of the genotype in males, and the frequencies of genotypes in females are obtained by Hardy-Weinberg principle to estimate allele frequencies. In one generation, the genotypic frequencies are at equilibrium when random mating is occurring. If the alleles are X-linked and sexes differ in allele frequencies, the equilibrium frequencies are approached over several generations, as male receives their X chromosomes from their mother only, whereas female receives an X chromosome from both mother as well as father. For genes with multiple alleles, the Hardy-Weinberg genotype proportions are obtained by expanding a multinomial expression. To calculate the allele frequencies from a number of a genotype, we count up the number of copies of an allele by adding twice the number of homozygotes to the number of heterozygotes that possesses the allele and divide the same with the sum by twice the number of individuals in the sample. By this, we can see that we can not only apply Hardy-Weinberg law to the genes located on autosome or the genes having only 2 alleles, but also to the sex chromosomes and to the genes which have more than 2 number of alleles. Now let's apply Hardy-Weinberg equilibrium using an example. It is possible to establish whether a population is in Hardy-Weinberg equilibrium for a particular trait. Consider a system with 2 alleles, capital A and small a, and 3 genotypes, capital A, capital A, capital A, and small a. Among 1,000 selected at random, the following genotype distributions are observed. That means, if we screen 1,000 randomly selected individual, we have found 800 individuals belonging to capital A genotype, 185 individuals belonging to capital A small a kind of a genotype, and 15 individuals consisting of capital A small a kind of a genotype. From this data, the frequency of allele for capital A, which is denoted as P, can be calculated by multiplying 800 with 2 and adding 185 to it, and finally dividing this sum with 2000. We have doubled the number of a sample because we are considering diploid organism. The frequency which has come using this formula is 0.8925. Likewise, we have also calculated the frequency for another allele, small a, denoted by letter Q, which comes as 0.1075. This is the observed allele frequencies calculated from genotype frequencies. Now consider what the expected genotype frequencies would be if the population is in Hardy-Weinberg equilibrium, and compare it with the observed values. The genotype AA, we have observed 800 individuals belonging to this particular genotype. From the frequency of allele, we know the expected genotype formula would be P square. So, if we square 0.8925 and multiply with the number of samples screened, we can have the expected genotype frequency for AA genotype, which is 796.5 in this case. Likewise, we can calculate the expected genotype frequency for heterozygous capital S small a individual and homozygous small a individuals. With observed and expected genotypic frequencies, now we can compute statistical analysis with the chi-square test to confirm and determine probability that the difference between observed and expected value is a matter of a chance or not. The chi-square is computed by the formula of summation of observed minus expected frequency square divided by expected frequency. We can apply the formula for all the three genotypes and obtain a value. In this case, the value which has come after applying the formula is 1.3357. The p-value at 0.05 is 3.84 and R value is 1.3357. This shows the deviation of expected from observed is not statistically significant. We can always find the allele frequency from Hardy-Weinberg law if we assume that the genotype in the population are found in Hardy-Weinberg equilibrium. This is how we test whether the population is in Hardy-Weinberg law or not. The table given in the next slide will show that how allele frequencies can be calculated for these two different hypotheses. For example, the O blood group under hypothesis 1 is a frequency of homozygous genotype at two loci, which can be shown by both small letters, that is AA and BB. That means both the allele are recessive in nature. The frequency of one homozygote is a square of the allele frequency. If we use Fx to indicate the frequency of allele X, the genotype A dash means a situation or a genotype in which either both the alleles are dominant, that is capital A type, or they are heterozygote but one allele is dominant, that is capital A and a small a. In other words, any genotype but recessive homozygous, that is small a. Since the frequency of the three genotype at one locus must sum to 1, we can write frequency of one genotype as 1 minus frequency of other genotype or 1 minus frequency of A square, small a square. Then the frequency of A-BB genotype is 1 minus frequency of small a square multiplied by the frequency of small b square. The tables given in this slide shows the calculation of allele frequencies under both the hypothesis 1 as well as hypothesis 2. You can see that the chi-square analysis obtained on the basis of difference between the observed and expected genotypes in this table. For hypothesis 1, chi-square is 43.52, whereas for hypothesis 2, chi-square is 1.6. Clearly, the chi-square depicts that the hypothesis of 3 alleles at one locus is a better hypothesis than 2 alleles each on 2 loci. Thus, genotypic frequencies sampled from a population can be used to distinguish between the two hypotheses for the genetic basis of blood groups and likewise for many other inheritances. In the previous example, we have seen that how Hardy-Weinberg equilibrium is used to see the change in the allele frequencies from one generation to another. By doing so, we can predict that how evolution has happened over the generations. We can also extend the Hardy-Weinberg equilibrium to know about the mode of inheritance. We can understand this by some example of ABO blood group system. As we all know that the Landsteiner proposed 4 types of blood groups, namely A, B, AB and O. But now the problem is to explain that how these 4 types of blood groups are inherited. That means to know their mode of inheritance. In this, allelic or genotypic frequencies can be used under Hardy-Weinberg equilibrium to propose the right kind of a model for their inheritance. We can assume 2 kinds of a hypothesis here for their inheritance. In one hypothesis, we can consider that these 4 blood groups are because of the 2 independent locus, each having 2 alleles, one dominant, another recessive. Whereas in the other hypothesis, we can consider only one locus with 3 different kinds of alleles. Out of these 3 different kinds of alleles, 2 alleles can be co-dominant on each other. However, they can be dominant on the another 3rd allele. So students, let us summarize this session. In this session, we have learned about the concept of gene pool. A gene pool can be described in terms of allele frequencies. A sexually reproducing population are only temporary assemblages of allele. The genotype breaks down each generation when the allele are passed from one generation to another through gamete. This happens to maintain the continuity from one generation to another. We have also understood in this module that the primary goal of population genetics is to understand the various processes that shape the gene pool. And the Hardy-Weinberg law is one such law which evaluates the effect of evolutionary forces on allelic and genotypic frequencies. The Hardy-Weinberg law requires a population which is infinitely large in size, in which mating is non-random and which is not affected by processes like natural selection, migration or drift. If no such event occurs, then there is no deviation between expected genotype and observed genotype frequencies. The Hardy-Weinberg law can be used to estimate the allele frequencies if the population is in Hardy-Weinberg equilibrium for that locus. For example, the frequency of the recessive allele will be equal to the square root of the frequency of the recessive traits. There are many factors which contribute to the deviation of Hardy-Weinberg frequencies. One such factor is inbreeding. Inbreeding increases the percentage of homozygous individuals in a population. Close inbreeding is even more harmful because it increases homozygosity and boosts the probability of deleterious genes. The lethal recessive gene will continue to combine and produce harmful homozygous lethal genes. Although inbreeding is generally harmful, but a number of inbreeding organisms are successful. The another force which leads to the deviation in Hardy-Weinberg proportion is migration. The amount of change in allelic frequency due to mitigation between population depends on the difference in the allele frequency and the extent of migration. Migration has two effects. In the first effect, it causes the gene pool to look exactly similar, and the second effect, it shows how natural selection and drift leads to genetic differences between the populations. The direction and magnitude of change in the allele frequency is also dependent on the selection proportion. Selection intensity and direction of the selection related to dominance of the allele. Directional selection leads to favoring of one allele over another. Over-dominant selection leads to the maintenance of both the alleles, and in under-dominant selection, the heterozygotes has the lowest fitness compared to homozygotes. It is important to keep in mind that the real population are affected by evolutionary forces, and thus evolution results from the combination of complex forces. The microevolutionary forces increases the genetic variation within and between population is mutation and migration, and the genetic variation within and between the population is decreased by genetic drift and natural selection. Thank you.